Thanks to the organisers for the opportunity to present this research. So most published biomarkers never make it to the clinic. And this article represents an attrition rate of about 99%. So some of the reasons for that are technical issues with assay development, challenges with clinical uptake, including demonstrating improvements over existing clinical practice, but also a major factor is that many biomarkers just do not generalize beyond the cohort they were initially assessed in. So as well as various other factors, this leaves us with an opportunity to improve. And one of the places we can start to do that is with the cohorts that we're working with. So an underlying hypothesis in this particular work is that if we integrate epidemiological, pathology, other observational variables, we can identify phenotypes by which we can then group patients and use those to then make better cohorts that allow us to do better biomarker discovery because the cohort has got more coherence. So there are two examples here. One on the bottom is saying, classify blue versus orange. It's quite simple. You could think of that as a clinical outcome. So on the left side, we have a mixed cohort where maybe the shape could perhaps represent some kind of lifestyle factor. And so the model with that data would end up learning to classify the shapes, and you would have an enrichment of the orange variable in the square and the blue variable in the circles. But if we can refine that cohort, then perhaps these confounded variables, these stars, can then start to come out and give us a better biomarker moving forward. So that's what you can see on the right. It's a more refined cohort. Underlying that whole sort of set of ideas is that the environment is affecting the molecular trajectories of the cancer. And it's well known that, for example, UV light causes particular transition mutations. Smoking is associated with a different mutational fingerprint around transversions and also impacts KRAS signaling. So, for example, in lung cancer, it's well known that the KRAS mutant cancers are enriched in smokers. I think it's about threefold enrichment. And then, of course, alcohol has a similar effect with DNA adduct and cross-linking and drives estrogen signaling, which is an important risk factor in breast cancer. Nutritional status is another example where we have a high nutritional status impacting important cancer pathways like IGF and MAP kinase. And of course, obesity itself is linked to a host of different diseases. And indeed, adipose itself is an endocrine signaling tissue that, for example, creates an inflammatory environment. So again, we're impacting through environment and lifestyle on the underlying molecular drivers that are pushing the cancer. So this is a figure adapted from Fard and Reagan. And you can see that kind of summarizes that idea that the environmental factors are then changing the networks that then lead to particular attractors. And those attractors, those disease attractors, might lead to a similar kind of gross overall phenotype, like, colorectal cancer, say. But those patients with those different networks are likely to have a different set of biomarkers that will allow you to discriminate between good and poor prognosis within that individual attractor. So that's the part of the hypothesis here. And I'm particularly interested in colon cancer in these series of projects. And this just shows the age standardized incidence rate for colon cancer. It's very important global cancer, of course. And you can see, again, this geographical distribution, which shows evidence for an impact of environment on the development of colorectal cancer as well. So, brief words of introduction. I'd now like to talk about a colorectal cancer resource that we have in Northern Ireland. I'll then talk about a toolkit for data preparation that we've developed and published. And I'll go on to some more ongoing work about phenotype discovery, where we're using some deep learning to try to pull out these phenotypes for better biomarker discovery. So the Northern Ireland colorectal cancer resource comprises about 660 patients from 2004 to 2008. They're split between stage 2 and 3. And the vast majority of patients that were in the Northern Ireland Cancer Biobank jurisdiction in that time period. So it's very comprehensive. We have 212 colon cancer specific deaths in that cohort in the same time period. Running up to 2013. So it's very rich because we have a lot of epidemiological variables as well as mutational and enhanced pathology from a variety of sources for this cohort, giving us 155 tabular variables, plus the images from H&E staining, And we very recently completed the collection of transcriptome data for this cohort, which will then allow us to do molecular stratification. So on to the data preparation. This toolkit called EHD Prep is available from CRAN. And part of the aim here was to improve data quality. Health data is notoriously problematic. So we're addressing all of these issues: accuracy, completeness, uniqueness, consistency, and also syntactic correctness, which is important for machine interpretability. So I would encourage you to look at the Gig Science paper, and please, let me know if you do end up downloading this from CRAN. I'm always interested in feedback. So this is now applying EHD Prep for quality assurance in the Cohort 661 data set. So one of the first things we're looking at is to standardize the data. So we're getting rid of missing values, cleaning zero entropy variables, doing some consistency checks. This is the completeness heat map that you can get from the package. You can see that post-processing, there's a lot more blue here, which shows that the variables have got more completeness. So we've been able to weed out some of the issues with the data from that point of view. Also, we look for consistency. So in this case, you can define rules that pair variables. Like here we have the end stage and the number of positive lymph nodes. So the dashed lines on this plot show the boundaries for the number of positive lymph nodes that define that end stage. And you can see on the right that some patients with the N2 are actually, the number of positive nodes are actually in the N1 classification. So that's not consistent. And that allows us to then go back to the clinicians and think about the data. Ultimately, we could potentially think about excluding those variables. But, in this case, we're able to resolve that and to go back and really check carefully about the quality of the data. We also have an information theoretic approach to evaluating variable merging. So here, we're merging these two input variables. And you can see on the top, this merging strategy, the information content in input variable 1 is higher than the information content that variable has with the merged variable. So that's the mutual information content. So there's information loss. So you can see that in the plot. It's a diagnostic to say, hang on a minute, that merging strategy has lost information. It's not working. So you can improve that. And this is an alternative merging strategy where we're not losing any information when we're merging the variables. And of course, it's good to merge the variables if you can retain all the information, because that reduces the redundancy in the data set and allows you to perhaps have greater completeness, where you have variables for one set of patients or another set of patients. You can then bring them together and get greater statistical power. So then there's a series of other steps that we would do with this in this quality assurance pipeline. Of course, you're looking at encoding, like one-hot encoding. We have QC summary reports. And ultimately, then you end up exporting the data for analysis. Additionally, we have an approach that we call semantic enrichment. And this is an ontology-based approach to allow us to reason at a higher level, a higher semantic level. For example, here, we're showing the colon. And there's all these different sorts of annotations on the left side and the right side of the colon that, you might not think, unless you're an expert, actually related to the left and right side of the colon. And it so happens in colon cancer that is an important prognostic factor. So if we can aggregate all these different variables, then we're getting more patients that are all in the left side group or the right side group. Then again, we can do better statistical comparisons with. We use Snowbed CT in this particular example. And here we're annotating the Colo 661 variables in blue to the Snowbed terms. And then these orange terms have aggregated that information. For example, this tree has heart disease at the top. So there's a lot of things in Colo 661 which are basically telling us about heart disease. So again, we get a better handle on that high-level concept rather than just looking at these very sort of low-level specific details which are characteristic of health records. So when we're doing that, of course, that means we create meta-variables. And we do that by aggregating with, simple numerical operations like sum or maximum or average or product, that kind of stuff, We found this quite effective. And this is the whole data set now. So the size of the nodes here is the page rank centrality. So you can see there's actually a lot of these orange variables, which means there's a lot of meta variables being brought out. In fact, 1,600 in total. So we believe that's really enhanced the utility of the data set. Okay, so that's the data preparation step, and now I'll just talk about the phenotype discovery in the remaining time. So we're using deep learning. This is a sketch of an autoencoder. Many of you will be familiar with this. we have the input layer and then compressions down to the latent representation or the embedding layer, and then it then expands out to the output layer. So we can measure the reconstruction performance, and we're trying to compress the data, and that helps us then later on, if we're looking for phenotypes, we can have a better representation of the data in the latent layer, rather than taking all the variables in the input layer. And we're particularly using a variational autoencoder where we're learning the distributions and then sampling from those distributions in the latent layer. So we have the mean and the standard deviation. And this also means that because of the sampling, it's a generative model, so we can then produce representative output that's kind of dreamt up by the machine. So the training, we started off with 1,807 variables. This is the tabular data. And then we ended up doing some feature selection on this to get the non-redundant variables. And whilst we've got the reconstruction loss, which is the quality of recreating the image, we also have the clustering loss term because we're looking to create phenotypes from this data. So therefore, we're clustering patients into different phenotypes as described by the input data. So that you can see the progression of the clustering and reconstruction loss on the training data. So we have 5 clusters that came out, and we used k-means plus. We found that outperformed DP scan. And, you know, whilst there's separation in these clusters, they're also kind of, you know, linking to each other in this 2D representation. The silhouette score is quite good, so, you know, we're quite happy with that. And then we dig into that. This is the SHAP values. We can see that some of the underlying epidemiological variables are contributing more to some of these clusters than others. And it's a combination of different variables that are contributing. So, for example, stage is contributing to several clusters, but alcohol is perhaps more important for cluster 4. We also have performance status, which is the fitness of the patient, and then social or personal history type variables in some of the other clusters. So it's always a combination of variables. We can see very clearly that there are kind of easy to understand phenotypes coming out with this process. So it's kind of worked, which is always nice. We applied the same or a very similar approach to the Cancer Genome Atlas. In this case, we did a very similar approach, except we didn't have a clustering loss function at that time. This was the work of a master's student. And instead, we used k-means plus after on the latent layer. And what we found was also 5 clusters. It was a very different set of input variables. A lot of it's about drug regimes. So when we then dig into that, or I should say it was, again, a good silhouette score. But now we're seeing things like, for example, KRAS mutated cancers with a particular therapy regime, which makes sense because KRAS cancers do have, you know, their own kind of therapeutic track. There was another one with again, a couple of different therapy regimes. Interestingly, there was a female enriched cluster, and there was a trend in that cluster for a higher level of genetic abnormalities. There's also a male enriched cluster and a trend for that cluster of history of all the malignancies, as well as a treatment regime that was statistically significant. And then the final cluster with, again, a different therapy regime. So again, you know, this is playing out, it's working. In this case, the clusters also show a survival relationship, although we're not necessarily particularly interested in that. We're more interested in drilling into the individual clusters, but that's also reassuring. So then going on to the imaging data, we did use CLAM to do the segmentation and patching, and this just zooms in to show some of the different sorts of patch sizes. We ended up going with a 64 pixel patch size because we're looking to try to zoom in onto individual cells or groups of cells. And what we found was initially that the first encoder that we changed, which is this one, worked very well for kind of non-tissue type images. So we decided to use it as an anomaly detection approach instead, to filter out and then to retrain. So this shows the data that we actually thought of anomalous or interesting, but then this and this were the sets that did well. So you can see that's very bleached and doesn't work very well. So we'd rather get rid of these images. So then we retrained the autoencoder, and we ended up getting clusters. Maybe 5 is the magic number. But we didn't put the answer in. We got 5 clusters in this case. And here you can see then, because of the variational autoencoder, we're getting decoder outputs that cluster together quite nicely in terms of image space. And just to kind of quickly summarize those, we have perhaps the non-tissue in cluster 2, and then more nuclei in clusters 3 and 4, and then this kind of striated or speckled set. I need input from a pathologist on this. I know we are talking to pathologists, ongoing work, but it's working quite well. So then to take that up to the whole patient level, each of these blue dots represents a whole slide image. and therefore the sampled patches out of that image, we've got 1,000 in each plot, and we can see that those shapes are quite different across patients. And so when we, just using a simple house source difference approach, we identified then 3 cohorts from these embeddings. So again, we're producing these image-based phenotypes, and then the next step then will be to integrate the tabulated data with the imaging data. So this is what I talked about, and I'd like to acknowledge the group, and in particular Tom Toner, who is a very talented PhD student who's just taken up a position in Surdan, and Rashi Pancholi, a master's student who works on the Cancer Genome Atlas, as well as the collaborators Helen Coleman, Paul Miller, and Thorsten Foster. Thanks for your attention.
We have time for a quick question, but if not, we can move. Please, you can come to your.
It's a really great talk. I also do a lot of machine learning for healthcare research in my PhD, so it's very
Can you come to the microphone?
Hi, yeah, thanks for the great talk. So I was wondering if you could provide a little bit more insight into how you form these clusters. For example, like alcohol was found, or like some of your features were found in multiple clusters, right? And your loss function was also, as your epochs went up, the clustering loss actually went up instead of going down. So could you talk about that a little bit more, please?
Okay, so under time pressure.
You can continue also.
Yeah. So in this case, you can see the clustering loss function is effectively plateauing. We implemented an early stopping criterion. So this is actually going beyond the end of where the early stopping happens. So we would stop, but this is the data that shows the longer time frame. And then, yes, so you have different features associated with different clusters. One of the issues there is that you, of course, have a range of values in each of the features. So some clusters may have higher values contributing to that cluster and other having low values, but it's still important to define the cluster. So briefly, hopefully that answers your question, but we can talk later.
Sure, let's chat later. Yeah, thanks.
Thank you very much.